carried with you while going sampling. And so we will discussing some of the sensors, colorimetric sensors that we have developed. Like we have developed a sensing system for chromium. This chromium is a heavy metal or metalloid. It present in environment in two oxidation state, like chromium 3 and chromium 6. Whereas chromium 3 is having nutritional benefit to human, but chromium 6 is toxic. So we have intended to develop a colorimetric diagnostic mechanism for detecting this chromium 6. So for this we have used enzyme substrate that is called TMB. This is a TMB, tetramethylene methyl benzidine. This TMB, it's because of presence of peroxidase enzyme. In the presence of hydrogen peroxide, it forms oxidized TMB. This TMB is blue color. You can see in the panel that this TMB is colorless, and if we oxidize or peroxidize with presence of enzyme complex, then form blue color complex. So this peroxidase activity, if we can mimic, so what we found that this chromium 6 is having a this peroxidase activity. It can mimic the activity of this peroxidase enzyme. So in the presence of hydrogen peroxide, this chromium 6 particularly can turn this TMB, which is colorless, to blue color. So with this mechanism, we have foresight that can develop a sensor for this chromium metalloid. But not only this chromium, other metals also have this ability. Like we can see from the panel that iron, copper, and some of other metals can have this particular property of peroxidase enzyme. So what we did to mask this other element, we did using this some chelating agent. So what chelating agent we have used is EDTA. See, EDTA is a chelating agent which can specifically bind cations. It's like iron, copper, it can chelate. Whereas this chromium is present in anionic state. So chelate, this EDTA will not chelate the chromium. So based this principle in the detection process, we have added EDTA, and we have found that mm, uh, using EDTA only chromium gives signal. You can see if we have EDTA and chromium, and then if we add this TMB and H2O2, it gives a blue color complex, and this complex having a linear range. This linear range, it range from 0.025 to zero point 0.5 ppm. So this range is quite high, and it gives a very practical sense of using this particular mechanism. And as far as WHO or US EPA concerned, the limit for drinking water, the limit of chromium 6 drinking water is 0.1 or 0.05 ppm. And the detection limit, whatever we develop in this particular mechanism, is 0.023 ppm. So we can use this mechanism in our particular situation, and we can develop a good sensor out of it. Next, we have also used some electrochemical sensor. We have previously used these colorimetric sensors where you can develop some color, and here this we have developed some electrochemical sensor, especially in collaboration with CDAC Kolkata, and also IIT Kharagpur has helped us to develop some of the mechanism. This is under some project NSF, ICAR NSF. They funded this project, and we can see this oxidation of chromium 6 to chromium 3 can also have some electrochemical signal, and using this electrochemical signal we can also detect chromium. Further, we can have, if we coat the electrode with gold and Neophene, then also it can improve the signal properties, and also it can enhance the lower detection limit, and also like 25 to 100 ppb for the chromium 6. Some new nanomaterials can also be used for electrochemical sensing of chromium, like cerium tungsten. This particular work has been done in IIT Kharagpur, and and they have validated this particular mechanism along with the real water samples, and they found a very good range like zero point 0.6 to 100 ppb chromium 6 it can be detected. So so what the purpose of doing that, these are the small mechanism. Beside this, there are many other mechanisms are already reported globally, but whatever our vision of this particular chromium uh, detection is that to quantify the chromium 6. I told you before previously that ICP-MS can detect the chromium, but this chromium, they cannot tell you that it is a chromium 3 or chromium 6. That's this specialty of this particular sensing system, that it can easily detect whether it is a chromium 3 or chromium 6. Only the chromium 6, which is toxic for human being, they can be detected using this particular sensing mechanism. So if we have this particular reaction mechanism or color development mechanism along with a small spectrophotometer, and go to the actual testing site, and we can and have the result with us. So now coming to the another dreaded heavy metals or metalloid like the arsenic. See this arsenic present in two state like arsenic 3 and arsenic 5. Arsenic 3 is highly toxic, whereas arsenic 5 is a little bit moderate toxic. However, the mechanism we develop in our laboratory is based on the arsenic 5. See, arsenic 3 is mostly present in groundwater, which is anaerobic situation. But when this groundwater comes in presence of oxygen, like in surface water system, they become oxidized to from arsenic 5. So if we detect the arsenic 5 also, we can easily predict the amount of arsenic 3 present in the system. And how we can do that, I'm just giving you a particular example. Like here we have used nanomaterial, like in previous chromium case, there we have not used any nanomaterial, but here we have used a nanomaterial, that is iron peroxide. This is iron peroxide, brown color liquid. And if we have taken the HR tame and have seen the this size of this nanomaterial using HR tame and other nano characterization techniques, and we found this the nano iron dioxide is having size is around 2.79 nanometers. And this interlattice space is very important piece of this nanomaterial, because where those this adsorbent will be adsorbed, like it's having 0.21. So there are various delta iron peroxide, alpha iron peroxide. These are being used for sensing technology and also for remediation technology. And in particular, the sense of that this nanomaterials also have the nanozyme characteristics. It means whenever I use this iron peroxide and instead along with TMB and H2O2, this will give this brown color. TMB and H2O2 will be a white color, and along with TMB, H2O2, and this iron peroxide, it will give this bright green color or blue color. So using this technique, we have used for this sensing of arsenic 5. The beauty of this iron peroxide, that it can absorb or adsorb arsenic 5 on its surface. So whenever arsenic 5 is adsorbed on its surface, it loses its nanozyme activity. So in absence of arsenic 5, it is having nanozyme activity to produce this green color. But in presence of arsenic 5, it loses its nanozyme activity. And this is a concentration dependent manner. So you can see this from 0.005 ppm to 5 ppm. In this range, it is having this log linearity or linearity response. But the problem along with is that arsenic 5 especially behaves very similar to phosphate. So our surface water or drinking water also have phosphate, and also if present, then it can interfere. And and we have seen that around 23% interference can come from phosphate. So if we have, but phosphate can be easily determined from our normal spectrophotometry method. And if we have this data, and if we minus it, the phosphate data, we can have this detection of arsenic 5. So but the presence, there are many reports there that this arsenic binding of arsenic is more faster or more competitive to than this phosphate. So with this particular mechanism, we have developed this sensing system, which can generate a green to colorless system. See, you can see here, if arsenic is present, then it is become colorless, and if arsenic is not present, it becomes clear, green in nature. So using that, we have developed this sensing system. I have some problem with my. Okay, so we have developed this sensing of arsenic, and you can see the detection limit is 6 ppb, around 6 microgram per liter. It can be detected, and this is far lower than WHO standard of water. There is a standard for at least around 10 ppb. So as per as WHO, the presence of the detection, I mean the MRL or permissible limit of arsenic is 10 ppb, whereas our this particular mechanism can detect arsenic at around 6 pp. So it is well below than the permissible limit. And as like the previous case of chromium, this ICP-MS instrument can detect total arsenic. It cannot detect the arsenic 3 and arsenic 5. So if we have this particular data, arsenic 5, and if we can minus the from total arsenic value, then we can have the precise data of arsenic 3. So using this particular mechanism, we can have the data of speciation. So the speciation means you can separately detect how much arsenic 3 or arsenic 5 is present in particular samples. So and we have tested this particular mechanism in water and also in fish also, because now from water this arsenic comes to biological organism like through plankton to fish, higher animals. It's called bioaccumulation or biomagnification, and ultimately reaching to the human health. So and this particular organism, this particular arsenic is accumulated in fish or any animal in the organic form. So like methyl arsenate or dimethyl arsenate, and these are non-toxic form. So if we analyze this non-toxic arsenic in ICP-MS, it will give the presence, it will show that the arsenic is present, but it will not show that non-toxic form of arsenic is present. But if we use this particular mechanism of this colorimetric method, then it will precisely says it is a inorganic form of arsenic which is toxic present in the samples. So and if suppose we have to test how much arsenic 3 is present, then we can simply oxidize the arsenic 3 using suitable oxidant, and it will give the total arsenic in the presence of total arsenic. Now coming to the other pollutants, like since it's a fisheries institute, we work a lot of fishes in and around the West Bengal, Assam, Orissa, and I have seen that there are a lot of occurrence of pathogenic bacteria. So these bacteria causes a lot of diseases in fishes, and Aeromonas veroni, Aeromonas hydrophila. There are in number of bacteria are present in fisheries aspect, and they are causing havoc around. It can even cause 100% loss in aquaculture field. So there was a challenge to detect, see, these bacteria can be detected through normal biotechnical tools like using PCR, but it is a lengthy process. You have to isolate the bacteria, then purify the bacteria, then process the bacteria using normal biotechnical process. So there's a challenge to develop this sensing of this particular bacteria using nanosensors. So here the MRE or molecular recognition element we have selected is aptamers. Aptamers are generally a small chain of nucleotides, either DNA or fragments or RNA fragments, which are having specific specific specificity towards particular analyte, especially a bigger, a larger molecule, or a bacteria or a virus. So these particular aptamers are identified or synthesized using salix. This is a particular depiction of some small depiction of this salix procedure, where a large DNA aptamers or RNA aptamer library are being used, and from a selection of this particular library, a single DNA or fragments, RNA fragments are selected. So you can Google this term salix, and you will have a total protocol how to prepare or identify those aptamers. However, globally, we could find that there is no report of this the aptamer for specific for this particular bacteria. So we have done our own salix, using the help of one research group at IIT Guwahati, and they have identified aptamer of this particular bacteria, and this bacteria aptamer we have used for sensing of this pathogenic bacteria. So what the principle lies behind that we have here, we have used gold nanoparticles. Previously we have used iron nanoparticles, which shows the nanozyme activity, but here we have used gold nanoparticles, which also shows nanozyme activity. So gold nanoparticle along with hydrogen peroxide and TMB, it produce blue color or purple color in nature, blue color coloration of TMB. So and if we absorb this aptamer on this particular gold nanoparticle, it inhibit the formation of blue color. Now, uh, what happens if we use this bacteria in this particular system? This aptamer will dissolve from this gold nanoparticle and attach to this bacteria, and this gold nanoparticle will be again recover this nanozyme activity and produce the blue color. See here, you can see, I can give you a schematic that this gold nanoparticle is having nanozyme activity, and if we have this aptamer on the surface of gold nanoparticle, it forms a nanozyme map. There is no nanozyme activity, but whenever this bacteria is present, this aptamer dissolve from this gold nanoparticle, and this gold nanoparticle again recover the nanozyme activity. So using this principle, we have develop a sensing system of this bacteria, and you can see this particular absorbance. I can see this gold nanoparticle, and gold nanoparticle TMBH2 gives a blue color, and whenever aptamer is adsorbed on the gold nanoparticle, this blue color formation, it will reduce. But whenever this bacteria is present, gold nanoparticle again recovers its nanozyme activity, and again this color forms. So, and this particular system is very much specific this particular bacteria, because this aptamer is only binds to this presence of particular bacteria or Aromonas verae. In presence of other bacteria like Aromonas hydrophila, we have checked, then Pseudomonas aeruginosa, then Klebsiella pneumoniae. So this blue color signal or purple color signal only comes in presence of Aromonas verae. So this particular sensing system we have developed for bacterial case. Now, coming to the next sensing system that I told you, in fisheries or aquaculture system, lot of bacterial problems are coming, and it can cause huge loss to farmers. So farmers tend to use lot of antibiotics. So starting from beta lactam group to tetracycline group to various new type of antibiotics, they are also being applied. So we started with tetracycline because, as far as Government of India is concerned, till now government has approved tetracycline group of antibiotics for fisheries application, but still farmers are still using a lot of other antibiotics, new generation antibiotics. So how to detect these tetracycline antibiotics? As here also we have adopted the approach of aptamers. So in this case, aptamers, we have selected aptamers from Google search or internet. There are aptamers already reported for tetracyclines, and we have collected that information and synthesized our own aptamer. and also you have used gold nanoparticle and its nanozyme activity. So the principle lies same here. So in the like in the previous case, in presence of this aptamer, the nanozyme activity of this particular gold nanoparticle inhibits, and whenever halides are present, like, the nanozyme activity of gold nanoparticle it recovers, and this blue color formation. But here we found a totally different approach. Previously, we thought that the color will be higher in presence of analyte, but we found that this the color is inhibiting. Why it is happening? um, that there's a high affinity of this aptamers towards gold nanoparticle, as well as this analyte. So what happens? This aptamer is attaching to the gold nanoparticle as well as this analyte. So I'm giving you a glimpse, like this is gold nanoparticle, and if we this is the nanozyme activity of gold nanoparticle, and this is aptamer in attached to aptamer, and the color reduce again if we present this tetracycline. And why it happens, we can see that this gold nanoparticle and aptamer complex is having still producing some blue color. But if the analyte is or tetracycline is given, it further inhibits the nanozyme activity of gold nanoparticle to produce a colorless system or nil color. So particular this mechanism we have developed uh, for this sensing of tetracycline, and you can see this it gives a log linear response. So it is not an actual linear system, it's a log linear system. If we have log in the x-axis, it will give a linear response. However, the problem lies in the binding of this aptamer to the analyte. See, what we have found, that this aptamer binds to the analyte in presence of particular binding buffer. So this binding buffer, so you can see, if the binding buffer is present as such in the reaction mixture, it also hampers the nanozyme activity of gold nanoparticles. So we have standardized the amount of binding buffer in this system, and we have found that if we use that 8 microliter binding buffer, then this nanozyme activity is perfect. And we have standardized all the reaction parameters and found that this particular linear range is 0.01 to 0.5 ppm. Within this range, it can detect the tetracycline, and the detection limit is 0.017 ppm. So this 0.017 ppm is much less than the prescribed limit or tolerance limit, that is zero point 0.1 ppm, which is set by European Union and FSSI. And so using this particular reaction mechanism, we have we can detect the tetracycline residue in water, and as well as in fish mussel. This particular actually is a output of our NSF biosensor project in collaboration with CDAC. Now we can all say that this is all about chemistry. How what about the sensing part, and how to take this particular mechanism to the lab to field. So our objective of this particular project is to develop a small device also, so that it can detect the color. So in that particular approach, CDAC has developed a sensing device. CDAC Kolkata, Dr. Sobik Pal, Shubhankar Mukherjee, they have developed this particular small device, and they have named the Imagine. This is a image-based instrument. In spectrophotometer is a your light passes through the sample, it gives optical density, transmittance, and reflectance, all those things. But here the color is being measured in lieu of like RGB, and based on this color, this gives some certain signals. And this is also a mobile based app they have developed. And if you give those data and standardize this particular algorithm, you can develop algorithm and develop a linearity in this particular app. And if you go for an unknown sample, it gives you a result. So these are the some of the results which are validated or developed in CDAC. There's a general CDAC for using chromium detection technique using that TMB what we had discussed earlier. And you can see that in this particular case, this LOD or limit of detection, limit of quantification also gives very low value, that is 0.12 ppb or 0.136 ppb. And and so this is a case of comparison between the UV spectrophotometer value and imagine. I told you that was a lab-based data, and this is this our developed imagine small instrument-based data. Around 25 samples, 27 samples have collected from river, Ganga river, and we have spiked, or some species are spiked and some not spiked, and this is kind of comparison. So this is the recovery of samples of chromium, and we found that around 100% or 99% samples your chromium can be recovered. And other than this chromium, we have also testing like arsenic, this tetracycline, then this pathogen bacteria. These are all still in. We are validating this result using those developed image based instrument. So regarding future perspective, what will be coming next? So see, science are always evolving, and people always want something new. So, what as far as new is concerned, so we can there are a lot of scope for developing sensors for emerging contaminants. So it can be like PFAS, it's a very new emerging contaminants, then antibiotics, some cosmeceuticals, and including microplastic. This is causing a havoc, and some of the sensors are already being reported for developing sensor microplastic. However, as far as fisheries is concerned, aquaculture is concerned, farmers are not bothered about this. Sensing of sensor of these particular systems like PFAS antibiotic. Farmers are more bothered on the more like production system sensor like dissolved oxygen, ammonia, phosphate, because these are the sensing system that make them aware or make them manage those fishery system more good way. Like most critical parameter in fisheries aquaculture is the dissolved oxygen and ammonia, but still now sensors are present of this particular system of these particular parameters, but those sensors are not at all in I mean, robust in nature. After maybe 6 months or 1 year, uh, they don't work properly because we have seen that, and they're also costly and high amount. Anyway, now this is all together from my side. I may have taken time, so some of the publication that we have based on laboratory studies we have did. We have a good number of publication on this. Whatever I have discussed in this particular presentation, all are being published, and you can see those publication. So with this, I thank you, CDAC and other associated organization for inviting me to give this presentation. I know this is a very short time to discuss all, but organization has told me to give only 15 slides and giving a time of I think 30 minutes. So I thank you all for have a kind patience for listening me. So I hand over to the moderator.